Okay, now here you seeing is the macula, lutea and fovea centralis. As I told you that the parallel rays of light, when they pass through the eye lens with they are passing to the principal axis of the eye lens, they strike straight behind on the opposite pole of the eyeball. So that's the center of the retina. So in the center of the retina, as I already told you, that the concentration of cones is more and that concentration of rods is less, right? So there is more clarity of vision. Now as you converge towards the posterior pole, you find that the layers of retina, they start thinning out and following a shallow curve or a depression, and that is around 5 to 6 millimeter in diameter, and the posterior pole of the here you see, I believe if those who are beginners, they have not seen this thing what I am talking is about. It's like this. Then talking is about this thing. The rays of light, when they converge here and they meet here at this point. So this is the point of visual acuity. This is the part of retina I am talking, right? And here there is a depression, and that's called macula of the retina, and it's called macula lutea also, because there is xanthophyll carotenoids type of pigments which give it a yellowish coloration when you see with the help of an ophthalmoscope in the retina. So as I was talking about this layers, can you see that at this point there is the thinning of the retina at this area called macula, right? And it is around 6 millimeters, so this you can see is around 6 millimeters in diameter. This is around 6 millimeter in diameter, this much. Take. But in further down, in the center of this, further down in the center of this, there will be an area here. This is even smaller, and this is around 1.5 millimeters. This area is called, it's a funnel shaped depression in the macula, in the center of macula, and that area is called fovea centralis. This is called fovea centralis. Okay, so let's study about this fovea centralis. To remember this, I think I need to give you an example. Like in marriages, people go to see the face of a bride, and the face of the, if to see the face of the bride, in some communities they are hidden, completely hidden. There are flowers hanging, strands of flower, then there are some silver foils things, then there are other sheets, other layers of veils. There's different layers of the veils covered on the bride's face, and they offer some tips or gifts to see the bride's face. So what is done, they have to reflect one by one the layers, 1, 2, 3, 4 layers, and ultimately the down, one gets to see the face of the bride. Got it? Just imagine the way we are sitting inside the vitreous chamber and you are trying to see the fovea centralis, right? But there are 10 layers. When you are facing the retina posteriorly, sitting in the vitreous chamber, there are 10 layers. So here what happens is you have to see, the fovea centralis. or fovea centralis, what I'm talking is, that means you are trying to see the cones. It's the area of the clearest vision, maximum bright light you appreciate at hit this point, and there are only cones rather. So to see the cones, you will have to reflect the layers one by layer. So one by one, when you'll reflect, actually you cannot reflect the layers of the retina, of course. What you can do is you can, heap them out, like push them to the periphery by like digging a hole, a shallowness, creating a shallowness. So what happens is this, the innermost layers of the retina, they will be shifted aside, creating a shallowness in the posterior pole of the retina, and that's called macula. And further you dig deep inside to further remove the layers, like 4, 5, 6 layers, from 10th, of course, you cannot replace, because 10th you cannot replace, because that of course is a transparent limiting membrane. So you leave the 10th, you push aside the 9th one, then you push aside the 8th layer, then you push aside the 7th layer, then you push aside the 6th layer, and you push the 7th layer, 5th layer, and then the 4th layer. So ultimately you will reach to this outer nuclear layer, and that is the layer of the cell bodies of rods and cones. That means you have reached to see the photoreceptors now. So in doing so, you can see what I was trying to show you is the layers have been shifted this way, right? So the layers, when they have been shifted this way, so what have you seen? That there's a shallowness created down there, shallowness created, right? So about fovea centralis, the important thing is that at this point you will find the rays of light will strike directly on the photoreceptors. The rest, all the layers have been shifted aside, and they have been heaped up on the lateral side of the fovea, or you can say even pushed or reaching to the reaching lateral to the macula. So this is the point where the light rays of light will strike directly on the photoreceptors. And what photoreceptors is? Pigment, of course. This will be the purpose of pigment epithelium, is to absorb the light. Then photoreceptors actually have 3 layers, if you think. One will be the layer of rods and cones. This is the layer I'm talking, is the layer of rods and here I'll be talking, because now I'm talking is about the cones only. So here you'll find only the cones, one thing. Then of course you will have the outer limiting membrane. This is the outer limiting membrane, right? Outer limiting membrane you will have. Then the outer nuclear layer will be there. This is the cell bodies of the rods and cones, right? So that's the limit which you can reach directly, right? So axons, remember at this point, because you have been digging the hole, so the axons of these cones, forget about the rods here, the axons of the cones will be shifted laterally, right? They'll be obliquely placed. Remember this thing. There's a very important thing about this. So the outer plexiform, this is the outer plexiform layer which I have drawn. That means the axons of the cones are not perpendicularly placed at the fovea centralis. This is a very important point. Remember that. The axons of the cones at fovea centralis are obliquely placed. I'll again be asking you this thing when I'll be comparing the histological sections of retina from the central retina and peripheral retina. Remember, this is one feature which will help you remember the identification. Okay. Then Yes, so this is like how much you will find. That means what are the layers you'll find in the fovea centralis? It will be the first, second, third layer, 3 layers. Okay, and the outer plexiform layer will be obliquely placed, will be pushed to the lateral sides. What else should I tell about this important thing, fovea centralis, the area of highest vision? There are only cones present here. And then you have, only and the density of cones is also very much here. Okay, so I think it's enough for this macula, lutea and fovea centralis. I believe you've understood, and let's move to the next slide. Okay, now you are seeing is optic disc, also called the blind spot, and Lamina cribrosa. So as I have already told you that if you see the eyeball from behind, let's say it's the right eye, right? So it's this point from where the optic nerve emerges, and it's this point here the fovea centralis is. Means this point here which I'm talking is this point from where the optic nerve emerges out, and that point is called optic disc, which is around 2 to 3 millimeters nasal or medial to the fovea centralis, right? If you see in other view, remember that optic nerve is not arising from the posterior pole. Right, so it's this, the fovea centralis here. This point is the fovea centralis, and this is the point which is called the optic disc, right? This is the sagittal section, this one is the this one is the sagittal section, and this one is the posterior view. Okay, so now here you seeing is a a transverse section passing through the optic disc. Now this point is also called a blind spot. Why blind spot? Because there is no photoreceptors here, though no rods and cones here, right? Because the nerve has to exit through this, the veins has to exit through this, and what has to enter is the artery to the retina, central artery to retina. So that will be entering through this, the veins and the nerves will be leaving through this point, that is optic disc, and that's why it's called a blind spot. Now look here, I could have zoomed it, but it's not zooming, I don't know how. So this area here you're seeing is the retina, right? This is the retina, this is the retina. Okay, so you have been told about all the layers. Now we actually focus upon the nerve fiber layer, which was the 9th layer, and nerve fiber layer is the central processes of the ganglionic cell layer, and ganglion cell layer are the second order neurons. Remember, there were three neurons along the pathway of this optic nerve, optic pathway in the retina. First was the photoreceptors, which they are of course modified neurons, but we don't count them as the first order neurons. They are receptors. Then the first order neurons in the optic visual pathway is the bipolar neurons. The second order is the ganglion cell neurons, and these are the second these are the axonal processes of the second order neurons, the ganglion cells, which converge. Now these fibers, they are actually converging, they are converging towards this optic disc, right? So these fibers are converging here of the optic disc, and when they converge, remember one important thing I told you before also, that the macular fibers will pass through the center of the optic nerve. Or you can say that macular fibers will reach towards the center of this macula. Now this point is called the macula. This I am talking is about the, sorry, not optic disc. This is optic disc. This is the optic disc we are talking about. Here these fibers which are converging from the entire retina. Remember that the fibers from the macula, they converge such that they are centrally from the macula, the fibers, they converge here at the center. The macular fibers, they pass towards the center of the optic nerve, while the peripheral fibers, they pass towards the lateral peripheral portion of the optic nerve. And how this pattern is maintained is I'll tell you further in my slide, but it is really important, as I told you, that the entire image which is formed on the retina, it is transported as such. The pattern is not distorted, the pattern is not disturbed. I told you the mirror inverted image from the object is formed on the retina, and that image is transmitted as such along the length of the optic pathway and is relayed there in the visual cortex. So macular fibers will stay in the center. Okay. Then there will be blood vessels here. I told you the veins also will be running here, and there will be arteries also running here. Arteries maybe like this, you can draw it. There will be central artery of retina. These all the structures passing through this. So this point of exit of nerve fibers is called optic disc. There is no rods, no cones, no photoreceptors, therefore it is called blind spot. Okay. Now think about how will they pass out, because this sclera, which is the outer fibrous layer, at this point it is having a sieve-like perforations. Okay, so this you can see is the vascular layer. They have shown is the choroids. Okay, this is the choroid, the vascular layer, and the beyond to that, this layer is the layer of sclera. Okay, so sclera at the area of the optic disc is having holes, having miniature minute holes, and because of that it's called cribrosa. It has holes, like it has perforations for the nerves to pass out, and that's called lamina cribrosa. Okay, so lamina cribrosa makes this point a little weak, of course. Structurally, if you find it's a little weak. And another thing, can you appreciate vessels? Of course, I have shown you here, not only the central artery but central vein of the retina, both of them will be passing to the center. And the important thing that you should have appreciated by now, can you see the thickness of the optic nerve here, right? And these optic nerve fibers, when they're emerging out like this, right, they're emerging out like this. Can you see the thickness of the optic disc, right? It's only this much at the lamina cribrosa, this much, right? So that means, and even further narrowed out at this much. So that means there is mechanical compression of the nerve fibers. Actually, it's a natural thing. I should not use the word compression. Rather, they are actually so tightly packed up at this point, like optic disc. So all those processes of the ganglion cells which are forming the axon, which are forming the optic nerve, which are rather emerging out of the eyeball in the form of optic nerve, they are getting so much packed up here And this has a very important significance, that being so much tightly packed up neurons of the ganglion cells which are emerging out of cribriform Lamina cribrosa, there are of course chances of ischemia. Okay, so because of tightly packed up nervous tissue, there are chances of ischemia at this point. And because it's been created by nature, so that also have a supplementation for this. And I'll tell you in my further lecture that there is a arteriolar anastomosic circle around this, so that it doesn't undergoes any sort of ischemia. So there were additional and a well perfused anastomosis at the cribriform Lamina cribrosa formed, and that is called arteriolar circle of Zinn. This is arteriolar circle of Zinn. Very important. So that, like, in case of any compression of the nerve, there shouldn't be any loss of oxygen and nourishment at this point. So there needs to have an additional enough supply of blood, and for that you have an and this is formed by short ciliary arteries. Remember, formed by short ciliary arteries. Short posterior ciliary. These are formed by short posterior ciliary arteries. Okay. So that was about the Lamina cribrosa, optic disc, blind spot. Now let's move to the next slide.